Hi, so in today's presentation we will be talking about proteins and all you need to know about them. So the first question is, what exactly are proteins? Proteins are large bimolecules or macromolecules. Their molecular weight is from approximately 10,000 to several million daltons. They are consisting of one or more long chains of amino acid residues. They are present in all living organisms. Proteins perform a vast array of functions within organisms. They are directly involved in chemical processes essential for life. They are the most diverse group of all biological macromolecules. Proteins differ from one another primarily in their sequence of amino acids, which is dictated by the nucleotide sequence of their genes, which usually results in protein folding into a specific 3D structure that determines its activity. They are also organ-specific, which means that their structure is dependent on the place where they exist. For example, proteins in muscles are different than proteins in brain. The most common elements of composition of proteins are carbon, oxygen, nitrogen, hydrogen, sulfur, and phosphorus. This composition is not the same as the composition of amino acids. This is because most proteins have various other molecules attached to their amino acid residues. Proteins fold into different 3D structures. Their natural shape is called native conformation. Some proteins can fold unassisted, but others require the aid of molecular chaperones. Their structure is usually discussed as a hierarchy of 4 different levels of structure. So the first presented structure is a primary structure. It is an amino acid sequence. Because the R groups that distinguish the amino acids play no role in the peptide backbone of proteins, a protein can consist of any sequence of amino acids. Any of 20 different amino acids might appear at any position. A protein containing 100 amino acids could form any of 20 to the power of 100 different amino acid sequences. This is an important property of proteins that permits great diversity. Secondary structures are regularly repeating local structures stabilized by hydrogen bonds. The most common examples are the alpha helix, beta sheet, and turns. Because secondary structures are local, many regions of different secondary structure can be present in the same protein molecule. Motifs are a pattern in a protein structure formed by the spatial arrangement of amino acids, or a stretch of protein sequence that mediates interaction between proteins. Many such motifs has been collected in the eukaryotic linear motif database. Many proteins are composed of several protein domains, for example, segments of a protein that fold into distinct structural units. Domains usually also have specific functions, such as enzymatic activities, or they serve as binding modules. The tertiary structure is the overall shape of a single protein molecule, the spatial relationship of the secondary structures to one another. Tertiary structure is generally stabilized by non-local interactions, most commonly the formation of a hydrophobic core, but also through salt bridges, hydrogen bonds, disulfide bonds, and even post-transitional modifications. The term tertiary structure is often used as a synonymous with the term fold. The tertiary structure is what controls the basic functions of the protein. The structure, formed by several protein molecules, polypeptide chains, usually called protein subunits in this context, which function as a single protein complex. In proteins composed of subunits, the interfaces where the subunits touch one another are often nonpolar, and they play a key role in transmitting information between the subunits about individual subunit activities. Only proteins with subunits exhibit quaternary structure. For proteins that consist of a single peptide chain, the tertiary structure is the final structure of the protein.
Chaperones are proteins that guide proteins along the proper pathway for folding. They protect proteins when they are in the process of folding, shielding them from other proteins that might bind and hinder the process. It turns out that virus proteins made inside the cells could not fold properly. Experiments have shown that an improperly folded protein can enter the chaperone and be refolded.
If protein's environment is altered, the protein might change its shape or even unfold completely. This process is called denaturation. Proteins can be denatured when the pH, temperature, or ionic concentration of the surrounding solution changes. Denatured proteins are usually biologically inactive. This method is used in food preservation. When protein's normal environment is re-established, after denaturation, a small protein might spontaneously refold into its natural shape, driven by interactions between its non-polar amino acids and water. This process is termed renaturation, and it was first established for the enzyme ribonuclease.
Proteins might be classified in three different ways. The first one is classification of proteins based on their composition. With use of this method of dividing proteins, we can group all of them into three categories, which are simple proteins that are constructed with only amino acids, for example hemoglobin, conjugated proteins that are constructed using amino acids and some non-protein substance, and finally derived proteins that are a product from either simple or conjugated proteins, and they are derived by physical or chemical means. The second method is dividing proteins by their functions. Using this one, we mark out 7 categories: transport, defense, enzyme catalysis, support, motion, regulation, and storage. The last method is based on the structure of the proteins. There are 3 categories in which we classify them: fibrous proteins, linear-shaped proteins insoluble in the water that perform the structural functions in the cell; globular proteins, either spherical or globular-shaped proteins, often soluble in the water, that form enzymes, antibodies, and hormones; and intermediate proteins, which are intermediate to linear and globular shape, soluble in the water. They are responsible for blood clotting. Now, when we know about protein structure, composition, a question arises: how proteins are made? Well, they are produced in a process called protein synthesis, in which cells make proteins. It involves 2 stages: transcription and translation. Transcription takes place in the nucleus, and during this process, genetic instructions from DNA are transferred to mRNA and then to ribosome in the cytoplasm. And that's exactly where another stage, translation, occurs. The code in mRNA is read in view to make a protein.
Functions of proteins. Proteins have diverse functions. Actually, there are so many functions of proteins that we could not list them all. However, we can divide them into 7 categories as mentioned earlier. The first one is enzyme catalysis. So basically, an enzyme is a substance that acts as a catalyst in living organisms, regulating the rate at which chemical reactions proceed without itself being altered in the process. And a lot of enzymes are 3-dimensional globular proteins which fit around the molecules they act on. That fit stress particular chemical bonds, which consequently facilitates the chemical reaction. The next function is defense. Some globular proteins' function is to recognize unknown microbes and cancer cells. The unknown cells are later on destroyed by white blood cells. Another function of proteins is transport. Many globular proteins transport small molecules and ions. For example, red blood cells contain the transport protein hemoglobin, which transports oxygen in the blood. Moreover, proteins can also support an organism by being responsible for many structures in our bodies: the matrix of skin, Ligaments and bones are only some examples of the structures formed by protein fiber. In addition, proteins play an important role in motion. Muscles contract through two protein filaments, actin and myosin. The sixth function of proteins is regulation. Hormones, which are small proteins, serve as intracellular messengers in animals. Proteins can also act as receptors for extracellular signals, and they have many other regulatory roles within the cell. The last category is storage. Calcium and iron are stored in the body by binding as ions in storage proteins in cells. Properties of proteins. As it was already said, proteins can differ by their properties, which are their shape, molecular weight, colloidal nature, denaturation, amphoteric nature, ion binding capacity, solubility, optical activity, hydrolysis.
Protein is a very valuable nutrient. Still, when it comes to diet, there are many different opinions on how much protein we need. The recommended dietary allowance in US is 0.8 grams per kilogram of our body weight, but many people think it should be more, just as in the chart you see. Now it is more than 0.8 grams per kilogram, so these rules aren't perfect because, as many studies show, amount of protein we need depends on many factors such as activity level, age, or state of health. Increasing protein intake may be helpful for people that are trying to gain muscle, and generally the more active you are, the more protein you need. Meat, fish, eggs, and dairy products are examples of whole protein products. If you're a vegetarian or vegan, you can make sure that your protein intake is correct by eating, for example, beans, peas, or rice. So don't panic, it may be a bit harder for you, but there are actually many plant-based products that contain protein. Another important thing when it comes to protein in diet is its quality, which is based on its digestibility and amino acid profile. Digestibility is so important because if you can absorb the protein you eat, it's practically useless. In general, digestibility of animal-based proteins is mostly over 90%, whereas plant-based protein digestibility is usually lower. It varies from 60 to 80%. As you can see on the chart below, the light blue is the animal-based proteins. So vegans and vegetarians need to eat more protein products than people that eat animal-based products to get the same amount of protein into their bodies. In general, proteins in food are digested by pepsin that breaks them down into smaller amino acids. Amino acid profile matters because all proteins are made from a mix of 20 different amino acids, and our body can only produce 11 of them, so we still need to get the other 9 through food because all 20 are essential for our health. On the chart, EAA stands for essential amino acids. These are the 9 that we can't produce ourselves. And as you see, animal-based proteins are more valuable to us in this category as well.
Proteins were recognized as a distinct class of biological molecules in the 18th century by Antoine Fourcroy and others, distinguished by the molecule's ability to coagulate or flocculate under treatments with heat or acid. Proteins were first described by the Dutch chemist and named by the Swedish chemist in 1838. The term protein to describe these molecules was proposed by Mulder's associate Berzelius. Protein is derived from the Greek word proteios, meaning primary in the lead or standing in front, prior to protein. Other names were used like albumins or albuminous materials. There are a few different fields of protein studies. Protein purification is a series of processes intended to isolate one or a few proteins from a complex mixture, usually cells, tissues, or whole organisms. Cellular localization, a process where a substance or cellular entity, such as a protein complex or organelle, is transported to and or maintained in a specific location within or in the membrane of a cell. Proteomics, a large-scale study of proteins. Bioinformatics, computational biology which involves the analysis of biological data, DNA, RNA, and protein sequences. Structure determination is a procedure by which the three-dimensional atomic coordinates of a molecule or biomolecule are solved using an analytical technique. Structure prediction and simulation. Develop sufficient mathematical models of proteins to computationally predict the molecular formations in theory. The activities and structures of proteins may be examined in three ways. 1. In vitro, which studies purified proteins in controlled environments. It is useful for learning how the protein carries out its functions. In vivo, experiments provide us with information about the physiological role of a protein in the context of a cell or a whole organism. In silico, studies use computational methods to study proteins.